Mendel was trying to figure out if two traits were linked together, or a gene, he didn't use the word genes, right? But he was pretty close. His theorems line up pretty consistently with what we know actually occurs in genetics. I mean, we learned, you know more from chapter 9 and 10 than Mendel did when he was performing his test crosses, but he was still able to come up with these. So he was trying to figure out, are two traits connected? And so he looked at two traits at once. You use the same rules as the smaller Punnett square. It's just the squares are expanded. We have more squares to deal with. Now, if our two crosses, if the individual dominant trait phenotypes is heterozygous for both traits, we're going to see every possible offspring. But when we cross two heterozygous parents for both traits, in what we call a dihybrid cross, we always end up with phenotypes in a ratio of 9 to 3 to 3 to 1. That 1 means 1 out of 15. We're going to see homozygous recessive for both traits. So for the most part, we're going to end up seeing heterozygotes. We use these, or used to use these in agriculture, in animals, purebred animals, to see what traits we end up with. Sometimes traits are linked together, etc. Let me just move into the test cross. Here I am giving the lecture. So what this means is, with the dihybrid test cross, you could be given two traits. So in this situation, we're talking about yellow and green seed pods and tall or short plants. So we know which one is dominant, which one is recessive. If we crossed a dominant for both traits parent with a recessive for both traits parent, that offspring, no matter what, is going to end up 100% heterozygous for both traits. But what Mendel did, and what these test crosses do, is they cross this first filial, this first generation, together, and then see what offspring we end up with, the potential we could end up with. The rules are the same. When we line up our alleles, you could only possibly have, in a sperm or an egg, one allele for each one. Remember, our chromosomes separate and they shift around, and so we're just putting potentials alongside here. So in this parent, it was heterozygous for the tall short trait, heterozygous for the yellow green trait. And so on this side, we just line up our possibilities in the sperm or the egg. So it's kind of like using that FOIL method in math. We could either have a big T, big Y, a big T, little y, or minuscule y, little t, big Y, little t, little y. That's one parent. Well, the other parent, because it's also heterozygous, is exactly the same. In the egg, we'd either have a big T, big Y, or a big T, little y, or a little t, big Y, or a little t, little y. Don't forget your basic rules. As long as you put your possibilities here, and you're only using one letter for each gene from then, it's just simply lining things up. So you could potentially see this offspring, this offspring, this offspring, this offspring. And your goal here is to be able to look at these genotypes and in your head know what the phenotypes are. Because if we see this, well, that must be tall, yellow, tall, yellow. What about this one? Tall, two recessive alleles must mean green. Tall, yellow, tall, green, short, green. So all this is telling us is the potential offspring that we might see. So we're going to see a lot more, that's where the 9 to 3 to 3 to 1 comes in, a lot more dominant for both traits than we would see recessive for both traits. So here I list the potential genotypes. Try to figure out if you can look at these genotypes and determine the phenotypes. And then practice with these dihybrid crosses. And let me know if there are any more questions about them. Some topics just need more explanation, and that's why I'm here. So reach out. Send an email to kbyers@mineralarea.edu. I can help give study tips, we can set up a phone call, or even a video conference.